to speak with Monty Python now for something completely different. We'll talk about immunology now. And as an introductory speaker to this, we really have probably the best person who can for this job. It's Hidde Ploegh from Boston Children's Hospital, who has given many of these lectures, introductory lectures at AI meetings, introducing the immune system and the structure and logic of the immune system. Scientifically, Dr. Ploegh has made a huge impact on immunology through his seminal work and very elegant work on antigen presentation by MHC molecules and by elucidating how pathogens evade the immune system. So this has been this kind of textbook knowledge now. He's also a member of the National Academy of Science and is a very distinguished immunologist. So we're really thrilled to have him here and introduce the immune system to us who know a bit about it and others who are probably novices in the immune system. All yours, and thank you very much.
Thank you. Well, as the day of this talk approached, it became clear to me that this is indeed a complex assignment. The immune system, such as it is a highly integrated system of many molecules and cells. And so what I'll try to do is touch on a number of topics that are of importance to immune recognition. In the beginning, I'll emphasize some areas, in particular in the area of innate immunity, where the movement of ions in small molecules is achieving is getting increasing attention. Then I'll move to some of the hallmark features of the adaptive immune system and highlight the unusual genetic events that underlie the creation of receptors, such as immunoglobulins and T-cell receptors. In the talk that follows, Leslie Berg will address the signaling properties of T-cells in greater detail, so I will not dwell on that particular topic in any great depth. And towards the end of my talk, I will introduce a set of tools, perhaps somewhat egotistically, but a set of tools that I think deserve wider acceptance. And especially in the area of ion channels, I think the use of small antibody fragments, so-called nanobodies, could have a profound impact on our understanding of how these channels work, with whom they talk, and what kind of methods one might deploy to interfere in their activity. So the host defense, I would like to divide this in three layers: mechanical and chemical defenses, which are omnipresent and act all the time. Think the integrity of the skin, epithelia, mucus secretions, tear fluid, and so forth. Many microbes and viruses have developed strategies to penetrate these mechanical and chemical defenses. And thereafter, we rely on what is called innate immunity to ward off these pathogens. And innate immunity makes use of receptors that are hardwired into the genome of the eukaryote. They recognize microbial patterns, molecules that are shared amongst bacteria, fungi, and many viruses. And the action is virtually instantaneous. It requires perhaps minutes to a few hours for the innate immune system to kick in. And as we'll see, the innate immune system comprises a large number of distinct cell types, many of the myeloid lineage, macrophages, dendritic cells, neutrophils, and so-called natural killer cells. They also make use of soluble mediators, proteins of the complement cascade, and small hormone-like molecules such as interleukins, interleukin 1, interleukin 6. These are all important in getting an inflammatory response going, and this has important protective value to the host. Now, importantly, the innate immune system, while it deals with probably 99% of invaders, isn't perfect, and evolution selects, as usual, for pathogens that can circumvent these innate defense strategies. And that's when we rely on the adaptive immune system to pick up the slack. And the adaptive immune system works in concert, in close consultation with the output of innate immunity. The main cell types involved are, again, dendritic cells for the acquisition of antigen. Macrophages do something very similar. And they, in essence, translate the antigens that they've acquired into a language that can be understood by T lymphocytes. B cells come equipped with an antigen-specific receptor on their surface, the generation of which I'll describe in some detail. And in order for an antibody response to unfold, a carefully orchestrated collaboration between T lymphocytes and B cells is essential. The kinetics with which the adaptive immune system kicks in is measured in a period of days to weeks. So to summarize, the defense system that all of us deploy consists of mechanical and chemical defenses, omnipresent and always active. Should these defenses fail, innate immunity kicks in on a scale of minutes to hours, and any invader that penetrates that barrier will be met with adaptive immune system components, and that takes a matter of days to weeks. Now, as far as the innate immune system is concerned, there is a huge amount of recent interest in small molecules that are sensed as evidence for harmful events. This complicated slide summarizes a few of the pathways that are important and in which transporters play key roles. So the innate immune system is geared to sense the presence of dead cells, DNA viruses, retroviruses, bacteria, and deals with these intruders based on shared components. Double-stranded DNA derived from reverse transcription of retroviral genome, self-DNA from mitochondrial, damaged mitochondria. These can be sensed by a cyclic dinucleotide synthase, which using ATP and GTP as the precursors produces these cyclic dinucleotides, which are then sensed by a molecule that is located on the endoplasmic reticulum. And its engagement with these cyclic dinucleotides results in activation of a signaling cascade that results in the synthesis of pro-inflammatory cytokines, interferons, and so forth. Microbes can also produce these cyclic dinucleotides. They gain entry into the host cell cytoplasm and activate the same pathway. So the exact mechanisms by which these cyclic dinucleotides are formed is beginning to be understood. We now know how these molecules are being sensed by sensors of the innate immune system, and we know a lot about the outputs. But this remains an area of intense investigational interest. Then we have a component of innate immunity that relies on activation of a high molecular weight structure called the inflammasome. If we start on the left of this slide, you'll see that gram-negative bacteria produce lipopolysaccharide. These lipopolysaccharides can activate caspases, leading to the conversion of an inactive precursor of a protein called Gasdermin D, a pore-forming protein in essence, that is upstream of activation of the inflammasome. The inflammasome contains leucine-rich repeats, a number of adapter proteins, and inactive proenzymes. And upon proper activation of these inflammasomes, the inactive pro-caspase-1 is converted to an active caspase-1. This is an enzyme absolutely essential for the conversion of an interleukin-1 beta precursor into its active form, which is then released from the cell by a non-canonical secretory mechanism, likely involving these pores formed by the polymerization of cleaved Gasdermin D. Now, there are many types of inflammasomes, and we don't fully understand exactly how each of them exerts their function, but NLRP3 is arguably one of the best studied members of this family. ATP is required for activation. Efflux of potassium, as we'll see in a moment, is essential also for activation of the NLRP3 inflammasome, always leading to the activation of conversion of inactive caspase-1 into the active form. Other members of the inflammasome sense double-stranded DNA. The AIM2 is an example of that. And there are bacterial toxins that modify small molecular weight GTP binding proteins, members of the Rho GTPase family. And again, this leads to activation of an inflammasome-like structure that contains a purine subunit required for induction of cell death. Now, as far as the NLRP3 inflammasome is concerned, potassium efflux, so reduction in intracellular potassium concentration, is essential. And there are a number of channels that can contribute to this loss of potassium. Bacterial pore-forming toxins are one example. The ATP-gated ion channel P2RX7, an ion channel that is activated by ATP, is another example. And the pannexin half channels likewise contribute to efflux of potassium. How exactly this reduction in potassium concentration activates the NLRP3 inflammasome remains to be understood in greater detail. But I want to emphasize the fact that this is an example of a response that will trigger inflammation. Flux of ions across various membrane components is important, and apparently many channels can contribute to this precipitous loss of potassium with concomitant activation of the NLRP3 inflammasome. There are yet other sensors of microbial products. Very well studied are members of the toll-like receptor family. I've diagrammed here in a cartoon the various members of this family. They recognize components such as peptidoglycan, lipopolysaccharides, yeast cell walls, bacterial flagelloproteins, and molecules produced by parasites. Upon engagement of these molecules, they contain leucine-rich repeats in their extracellular domain for ligand recognition. Downstream signaling elements are engaged, and this leads to activation of similar transcription factors to the ones I've already mentioned. These, again, induce the synthesis of pro-inflammatory cytokines, including TNF-alpha, interferon gamma, IL-6, and so forth. Yet another important part of the innate immune system are proteins of the complement cascade. This is a family of proteins conceptually similar to proteins involved in blood clotting. They are, in essence, proteases that act in cascade fashion, causing an amplificatory cascade, resulting in activation of the final components of the complement complex. We distinguish three different pathways of complement activation, one of which requires the presence of immunoglobulins. I'll return to that in a little bit. But as far as innate immune recognition is concerned, yeasts that contain mannose-rich cell walls can activate mannose-binding lectins, which in turn activate the MASP1 and MASP2 serine proteases. And these are upstream of complement proteins that will ultimately inflict cellular damage. There is an alternative pathway of complement activation that relies on ill-defined properties of microbial surfaces. And these activated complement components likewise converge on one member of this cascade called the C3 protein. The C3 protein is capable of forming covalent adducts with both hydroxyls and lysine residues. And upon activation or covalent decoration of surfaces with these complement components, subsequent partners in crime are recruited, the C5 through C9 proteins, and they together form a pore called the membrane attack complex, by which, again, ions can leave the cell, leading to cell death. So this complement cascade is important because it is abundantly present in the circulation. Any invading microbe that presents a proper surface will be a target for complement activation and can thus be eliminated. Equally importantly, the covalent modification of these microbial surfaces tags them for recognition by cells of the phagocytic system. So proteolytic fragments derived from the C3 and the C5 components serve as chemoattractants. They attract neutrophils primarily. The C3 fragment that decorates the protein surfaces covalently is the element recognized by complement receptors on phagocytic cells, and this assists in acquisition of antigen by cells of the adaptive immune system. So let's turn to the adaptive immune system. I've told you that the kinetics with which these responses unfold occur on a timescale of days. And this graph details some of the components that are considered part of an adaptive immune response. In the case of a virus infection, and that's the example we deal with here, very rapidly type 1 interferons are produced. These are the small hormone-like substances that are produced as a consequence of engagement of innate immunity. There's a cell type called natural killer cells that are endowed with receptors that can distinguish between virus-infected cells and uninfected controls. They come up very rapidly, but that may not be sufficient to contain the virus infection. As you'll see in the green line, the virus titer in my hypothetical example continues to increase. It is at this time that the adaptive immune system kicks in. The first members to show up on the scene are virus-specific killer cells, or CTLs, cytotoxic T lymphocytes. They rapidly expand and drive down the virus titer in the organism. Slightly behind is a wave of B cells that are activated. They begin to produce antibodies, and it's the combination of these killer T cells and antibodies capable of recognizing the virus that ultimately drive down the virus titer to levels below detection. One of the interesting properties of the immune system, which I'll not revisit later on, is a feature called memory. The immune system, once activated, returns to a resting state, but it retains memory of the antigens that it has experienced. So upon re-exposure of an organism that's already experienced the virus infection, if that same virus were to recur, what we see is a much accelerated adaptive immune response. The antibody titers go through the roof. Cytotoxic T cells are activated from a memory component, and therefore, upon this secondary exposure, virus titers usually don't rise to the level seen in the primary infection, and they drop down to baseline levels much more rapidly. Now, the immune system is highly organized. In essence, cells and molecules of the immune system occur in the circulation. And in order for them to survey tissues and events that occur there, they must also be able to leave the circulation and take up residence at sites where their action is required. The circulatory system sees to it that these molecules and cells are distributed throughout the body. There are organs called secondary lymphoid organs in which lymphocytes take up residence, and that is really where immune responses are initiated and sustained. While it's common in science, especially in human immunology, to take human blood as a surrogate for the human immune system, that is not entirely correct. All of the action occurs in these secondary lymphoid organs. It's the positive hydrostatic pressure exerted by the heart pumping that allows fluids to leave the bloodstream and permeate the tissue. And this liquid is then returned via the lymphatic system and filtered through lymph nodes before it is returned to the circulation. So the secondary lymphoid organs really act as filters through which materials acquired in the periphery are sampled. And it is at that location that immune cells, such as T cells and B lymphocytes, make their first contact with antigen. The family of antigen-presenting cells plays an extremely important role in this whole story. Just below the skin and below all epithelia is a dense network of specialized cells that carry all the molecules required for initiation of an immune response. These are the so-called professional antigen-presenting cells. Most of them are also classified as dendritic cells. These are the guys that make first contact with invaders. They contain all of the receptors required for activation of both innate and adaptive immunity. And as such, they are essential players in getting an immune response started. Now, the lymphocytes themselves are cells of hematopoietic origin. There's a pluripotent hematopoietic stem cell, which differentiates across all hematological lineages, including red blood cells, macrophages, dendritic cells, lymphocytes, and so forth. So although they all derive from a common precursor, at some point they differentiate into specialized cells called B lymphocytes and T lymphocytes as far as adaptive immunity is concerned. It's the B lymphocytes that make the antibodies, also known as immunoglobulins, But in order for that to occur, they require help from T lymphocytes, and how that happens will be described in a moment. Now, the antibodies that most of you are familiar with come equipped with a number of desirable properties, both from a fundamental biological perspective, but also from a biotechnological point of view. On the right, you'll see the ability of antibodies to engage the complement system reflected as part of an anti-tumor response. We know that antibodies can neutralize viruses, so that's another important function of antibodies, the ability to bind to a pathogen and, in essence, disarm it. Then we have on the left bottom, the ability of an antibody that is tumor-specific to engage through its conserved constant portions a cell type of the innate immune system called natural killer cells. And it's upon engagement of this conserved constant region of the immunoglobulin, once bound to a tumor cell, that this antibody-dependent cell-mediated killing can occur. I've already told you that components of the complement system can covalently decorate a foreign substance and facilitate its acquisition by macrophages. Well, antibodies can do exactly the same thing. If you have an antibody that binds to a tumor cell, then macrophages with receptors that recognize the conserved portions of immunoglobulins can capture these antibody-decorated tumor cells, engulf them, and in essence, kill them. And then, of course, therapeutically, we use antibodies as cancer therapy. We can use them as imaging agents. And this is why, in biology more generally, antibodies assume pride of place. Now, if we look at the structure of immunoglobulins, one quickly realizes they come in different shapes and forms. I've diagrammed here a few of the examples that are present. On the left, a pentameric version of a molecule called IgM. I don't think we should waste too much time on nomenclature. Suffice it to say that immunoglobulins occur in different flavors depending on the conserved constant regions they come equipped with. The pentameric IgM molecule should be distinguished from the dimeric IgA molecules shown in the middle. This is a protein found in mucus secretions and tear fluid and serves to protect epithelia from bacterial and viral invaders. On the bottom right, a molecule that those of you who suffer from hay fever and other allergies might be familiar with. This is the immunoglobulin E. Again, it's the conserved portions of the constant region that endow this molecule with its nasty properties. Now, if we look at a typical IgG molecule, it is composed of two identical heavy chains and two identical light chains. The two light chains, the light chains and the heavy chains, each have a variable domain located at the tip of the molecule and a conserved constant region, which is at the base of this Y structure. Then the conserved portion, that is the stalk on which these two arms of the immunoglobulin molecule sit, is referred to as the Fc portion. This is this conserved region recognized by receptors on macrophages and K cells and so forth. Antigen binds at the very tip of the two arms. The regions through which the antigen binds are referred to as complementarity determining regions, and they, using the standard rules of protein-protein interactions, create a surface that's perfectly complementary to that of the antigen to which the molecules bind. And the buried surface that results from this tight interaction accounts for the often very high affinity with which immunoglobulins can bind to their targets. Picomolar binding constants are not exceptional. The specificity of these antibodies is truly amazing. Now, if one considers the task of the adaptive immune system, which is to defend us against rapidly evolving pathogens, it stands to reason that pathogens with short division times have a chance to accumulate mutations that allow them to escape from the adaptive immune system. And spectacular examples, of course, include the influenza virus, HIV. These are viruses that rapidly change their antigenic makeup such that they can escape from immune recognition. In essence, the adaptive immune system is always a few steps behind the pace with which these pathogens acquire new mutations. And so that remains an issue that we need to deal with, not just with flu and HIV, but as we're now learning, probably also with SARS-CoV-2 at some point. If one looks at the type of variation that's diagnostic of these immunoglobulin variable regions, one notices that this variation is concentrated in predominantly 3 regions called complementarity determining regions 1, 2, and 3. And it is these 3 regions that find themselves at the very tip of the variable region with which they interact with the antigen. And so the question has been asked, how is it that these immunoglobulins can display such massive diversity in the face of the universe of pathogens that they need to deal with? If one looks at the diversity of immunoglobulins in a typical mammalian species, it's been estimated that there may be as many as 1 million distinct immunoglobulin molecules, each of discrete sequence and specificity. And if you were to calculate how that diversity is encoded in the genome, you would very quickly conclude that it's impossible to do so as germline genes, because you would expend all genetic space, if you will, by encoding immunoglobulins. And what's now part of textbook knowledge, of course, and many of you, I'm sure, are familiar with this, is the method by which immunoglobulin diversity is created. And this is done through somatic gene rearrangement. It turns out that the immunoglobulin heavy and light chains are encoded by disparate genetic elements that only in B cells rearrange to form a functional receptor. On the top, I've diagrammed a small number of variable region genes, some segments called J segments that will serve to connect the variable region to the constant region, and a single constant region. This gray dot is an enhancer sequence, and the process of rearrangement serves 2 purposes. First, by juxtaposition of a variable region with its joining segment, a complete functional variable region of the light chain is created. But promoters upstream of this newly generated VJ combination are then brought within controlling distance of the enhancer, allowing transcription. So the germline configuration is, in essence, transcriptionally silent. Once rearrangement occurs, we have both a functional immunoglobulin gene and a transcriptionally active unit. The heavy chain genes are even more complex. They contain a battery of variable region gene segments. They contain a constant region gene segment, in fact, several, and interposed between them, diversity elements referred to as Ds, and joining elements referred to as J. And again, by a process of sequential gene rearrangements, the V, Ds, and J are properly juxtaposed. This brings the promoter of the newly generated variable region of the heavy chain within controlling distance of the enhancer, and that allows this transcriptional unit to be active. So I like to compare this to pulling the handle on a molecular slot machine. For the heavy chains, we have V elements, D elements, and J elements, each of unique sequence. And what the immune system does in the course of B cell development is pull the handle of this machine. The wheels spin, a V, a D, and a J are selected. And this is how you can create, from a limited set of genetic elements, a massively diverse repertoire. What I've diagrammed in this cartoon is a slight refinement on that theme. This is a cartoon of how the heavy chain genes rearrange. Ds and Js are brought together first, but as the rainbow color indicates where the Ds and Js come together, the process of genetic rearrangement introduces some element of imprecision, which further expands sequence heterogeneity precisely where Ds and Js come together. The same event occurs when Vs are brought in juxtaposition with the newly rearranged DJ element. And so at the V to D junction, once again, we have junctional imprecision. Nucleotides may be added and deleted, accounting for yet additional diversity. So looking at this particular scheme, one begins to appreciate that diversity of the immunoglobulin subunits is created by somatic gene rearrangement. Inherent in this rearrangement process is some imprecision that expands sequence diversity exactly at those positions in the immunoglobulin chains where antigen recognition occurs. And then finally, the active rearrangement ensures that the promoter of the immunoglobulin genes in question become transcriptionally active through juxtaposition with an enhancer at the appropriate distance. What I haven't told you is that these immunoglobulins occur in two distinct forms, a membrane-bound form and a secreted form. The primary transcript of your typical immunoglobulin heavy chain contains a fourth exon called cmu4. This is one of the exons of the IgM constant region, followed by a sequence element that would normally specify the soluble version of this IgM molecule. Downstream of this cmu4 exon is a polyadenylation site followed by two transmembrane exons and a second poly-A addition site. Transcription runs through the entire block of exons diagrammed on the top, and it's this differential polyadenylation that allows the cell to make a choice between the production of a secreted IgM, this is when the proximal poly-A site is used, and when the distal poly-A site is used in the course of mRNA processing, a splicing event removes the little piece that accounts for secreted IgM and instead builds in these two transmembrane exons. So a single gene encodes both the soluble version of these immunoglobulins and the membrane-bound version. And this is, in biology, I think, a unique example where a single gene encodes receptors of different structure with which will execute different fate decisions. So if we look at how this process occurs in the course of B-cell development, the first event that occurs is to rearrange the gene that encodes the heavy chain of the immunoglobulin. It is a membrane-bound version. It is expressed in association with so-called surrogate light chains that do not contribute to antigen specificity. And the result is the formation at the cell surface of what is called the pre-B-cell receptor. This structure drives expansion of newly generated B cells. That is to say, every single B cell that successfully underwent this heavy chain rearrangement displays at its surface this receptor. It allows for clonal expansion. And this, in essence, safeguards any successful rearrangement that has been made. As these B cells differentiate, they switch off the synthesis of these pseudo light chains, so these surrogate light chains, and now begin rearrangement of the light chains. And this is, again, a random process. So a given heavy chain has no prior knowledge of the light chain with which it ends up. Once the light chain has rearranged, it becomes part of a fully functional membrane-bound B cell receptor, and that creates the B cells that can lie in wait until they meet an antigen for which they are specific. So what I want to highlight here is the fact that a single gene, in this case the mu chain, encodes a receptor that executes a fate decision. This is the pre-B cell receptor. It drives expansion of any B cell that successfully underwent gene rearrangement. And by subsequently recruiting the rearranged light chain, functional B cell receptors are created that can bind antigen. As I've told you, the immunoglobulin mu chain can be processed in such a way that it generates either the soluble released version of the immunoglobulin or the membrane-bound B cell receptor. And this is, again, a unique situation where a single gene encodes a membrane-bound receptor for activation and with signaling properties. And I think Leslie Berg's talk will address some of the signaling events that typify these antigen-specific receptors. And it can create a released soluble version that can act at a distance from the B cell that produces it. So how did this T-cell, B-cell collaboration take place? And this introduces the T lymphocytes to the story. We've seen that the innate immune system can equip phagocytic cells with the ability to acquire antigen through a variety of germline encoded receptors. Phagocytosis results in delivery of this antigenic material to endocytic compartments where the acquired materials are digested and proteolyzed into peptides. This is called processing of antigen. And we'll see that the fragments that result from these processing events are displayed on the surface of the antigen presenting cell in complex with products of the major histocompatibility complex. And it's these MHC peptide complexes that are recognized by T lymphocytes. This is an X-ray structure of a typical class 2 MHC molecule. It consists of two subunits, the green one, the alpha, the blue one, the purple one, the beta subunit. The two subunits together form a deep cleft into which these peptide antigens can be accommodated. And what the T cell receptor for antigen recognizes is the composite surface generated by the two subunits of the MHC complex and the peptide that's bound in the peptide binding cleft. There are two forms of MHC product, class 1 and class 2. The preceding slide shows you the two-chain class 2 version. These class 2 molecules are primarily centered on sampling antigen acquired by endocytosis. There is a second class of MHC product called class 1 MHC products. The central one here, HLA-A2, would be the typical example of a class 1 MHC product in the process of presenting antigens. I made this slide to emphasize the fact that these MHC products are part of a multi-gene family, each of which encodes a protein product with quite distinct functions, which ranges from lipid binding to FC portions of immunoglobulins that are recognized to the binding of transferrin. So TB collaboration requires acquisition of antigen also by the B lymphocyte. As you'll see, the B lymphocyte binds antigen to its B cell receptor. This complex is internalized. The bound antigen is digested and presented as peptides via these class 2 molecules present on all B lymphocytes. And that is what the T cell recognizes. It's important to distinguish between the T cell epitopes, which may be internal to a protein and require liberation by proteolysis, and what the B cell sees, which is of necessity a fragment of the protein that is exposed to solvent. I'll skip this slide and go to the collaboration between these T cells and B cells in the case of a virus-infected situation. I'll use this slide to illustrate the concept of restriction of recognition by products of the major histocompatibility complex. So in the above example, we have a mouse that's infected with a virus. Of the many B cells that could potentially recognize the virus, only a single one meets the proper criteria, and it seeks to expand and make immunoglobulins that can neutralize the virus. But in order for that B cell to be able to do so, it requires assistance from the T cells, as diagrammed in the preceding slide. And in essence, these T cells are specific for the same antigen. The recognition of these infected cells is such that uninfected cells are, of course, spared. The T cells that are charged with recognition of virus-infected cells ought to distinguish between healthy, uninfected cells and the infected cells. And moreover, they can also distinguish between different viruses, even if they're closely related. So a T cell that recognizes this year's flu antigens might not recognize what comes down the pike next year. And it's these T cells that can then collaborate with B cells of the appropriate specificity. The processing pathways are important to understand. In the case of the class 2 pathway, endocytic events are key. Endocytosed antigen is destroyed. Peptides are presented via class 2. We can also utilize the autophagy pathway, in which components of the cytoplasm are sequestered in autophagosomes and delivered to phagolysosomes for presentation via class 2 molecules. The pathway of assembly of class 2 molecules is understood in great detail. It is in essence a partner subunit of the class 2 molecules, the so-called class 2 associated invariant chain that delivers these class 2 molecules to the proper endocytic compartments and orchestrates the process in great detail. So these slides will be available. I can't spend all the time to walk you through the details, but suffice it to say that the pathways that lead to class 2 restricted presentation are extremely well understood. Class 1 molecules, also seen by T cells, focus on proteins present in the cytoplasm. It came as a surprise at first that these killer T cells seem to focus on proteins that aren't even displayed at the cell surface. A foreign antigen, for example, a viral nuclear protein, could be recognized as foreign and or misfolded. It becomes a substrate for ubiquitin conjugation and proteasomal proteolysis. And the products of proteasomal proteolysis, short peptides, are generated in the cytoplasm, where they may be further processed by aminopeptidases. These peptides must reach the peptide binding portion of the class 1 molecule, which is located in extracellular space. So in order to overcome this topological hurdle, there is an MHC-encoded peptide transporter called the TAP complex that's charged with delivery of peptides into the lumen of the endoplasmic reticulum. And there, in a complicated song and dance with a variety of chaperones, peptides are loaded onto the class 1 MHC product, which is then delivered to the cell surface for inspection by these killer T cells. The T cell receptor you'll hear about from Leslie Berg is, again, a complicated receptor tyrosine kinase-like structure that activates a phospholipase that cleaves PIP2, generating phosphoinositides that target the ER for calcium release. We heard about the channels that are involved in this process, the aura-I channel, the crack channel, the STIM1, STIM2 proteins, and I'm sure Leslie will revisit this, but this is why signaling via these antigen-specific receptors is an important area of pursuit for those of you who are interested in channels and transporters of various types. The crystal structure of the peptide transporter has been solved by Robert Tampe and colleagues, and it reveals the architecture of a typical ABC type transporter. It turns out that these transporters are favorite targets of viruses that make small proteins that block the channel through which peptides must flow into the endoplasmic reticulum. And so these TAP peptide transporters, in conjunction with the viral inhibitors, are a really nice example of this continuing chess game between pathogens on the one hand and adaptive immunity on the other. Now, in closing, a few words about tools that I think are underexplored as far as channels are concerned. And these are nanobodies, well known to structural biologists. Camelids, that includes alpacas, llamas, dromedaries, and so forth, in addition to making the 2-chain immunoglobulins about which I spoke, make heavy chain only immunoglobulins. So on the left, you see a 3D rendition of a conventional immunoglobulin molecule. In the middle, a version that lacks the light chain, unique to the camelids. And on the far right, the recombinantly expressed variable region of these heavy chain only antibodies, so-called nanobodies. These nanobodies have served as crystallization chaperones, and pretty much everything we know about G protein coupled receptors owes a debt of gratitude to nanobodies that were used to co-crystallize many of them. Their small size results in short circulatory half-life, superior tissue penetration, and they're very easily expressed recombinantly and modified with markers of choice. So for single molecule fluorescence studies, for creation of antibody drug conjugates, these are really quite amazing tools. They're made by immunization and phage display of the variable regions of the heavy chain only antibodies. This is a standard procedure that can be produced in pretty much any lab, provided you have access to these fuzzy animals. And we have used camelid-derived nanobodies to interfere, for example, in inflammasome activation. By making nanobodies that recognize the adapter centrally located in the NLRP3 inflammasome, the ASC protein, we can block activation of the inflammasome, block activation of caspase, and block release of interleukin-1. As I told you, these nanobodies serve as crystallization chaperones. We now know exactly why the nanobodies against this ASC adapter interfere with inflammasome activation, And this is by no means the only example. Finally, these single domain antibodies can be modified site-specifically using enzymes that recognize motifs deliberately engineered into these nanobodies. And these can be used for imaging purposes, both non-invasively in living mice, but also in correlative light electron microscopy to obtain highly resolved images of cell types of interest. The strategy we use is, I think, well known to structural biologists. Our nanobody is called the target protein of interest here. We modified with this 5 residue sequence motif diagrammed in single letter code. This motif is recognized by an enzyme called sortase A to form a thioacyl enzyme intermediate. And by feeding into the reaction a short synthetic peptide to which one may append any payload of interest, one achieves site-specific stoichiometric modification of the protein of interest with the moiety of choice. So we've done this with biotin, fluorophores, carbohydrates, radioisotopes, and so forth. As long as you can make this synthetically, you can affix any probe of interest to your favorite target protein. Now let me move to the final example that I show. So here we compare a thick brain section stained with a nanobody that recognizes the YFP protein. On the right, you see this brain section stained with a conventional anti-GFP protein colored in red. The neurons are expressing YFP under control of the Thi1 promoter, so every single neuron is green. And you'll appreciate that the conventional full-sized immunoglobulin barely penetrates the tissue section, maybe 1 micron deep. This was done in collaboration with Jeff Lichtman at Harvard. The nanobody, on the other hand, penetrates at least 50 microns into this tissue section, allowing one to arrive at 3D reconstructions of cells that span a considerable depth in these tissues. And because we have nanobodies that recognize markers on microglia, on endothelial cells of the circulation, glial fibrillary acidic protein in astrocytes, one can then arrive at a 3D reconstruction of a block of tissue. And as this 3D rendering shows, for the first time, we get a complete 3D reconstruction of microglia astrocytes with all their branches. You'll see in a short while neurons appear. The microglia retains its morphology perfectly. And we see evidence of synaptic pruning revealed by the presence of synaptic vesicles acquired by these phagocytic microglial cells. So in purple here, a neuron. One can look at neuronal interactions with the synaptic vesicles in place. And so nanobodies can then be used to arrive at these highly resolved structures of tissues that would otherwise be almost impossible to image at that depth. So in view of the fact that ion channels and transporters have been looked at by structural biologists, I would suggest that it might be a highly profitable enterprise to generate nanobodies against these ion channels for the purpose of selectively modifying them, changing their properties, connecting them to other proteins within the membrane in which they reside. To my knowledge, the P2X7 ATP-gated ion channel is one of the few for which nanobodies have been generated. And given the ease with which this can be undertaken, I think this would be a very interesting point of intersection for immunologists and physiologists interested in transporters and channels. On that note, perhaps I should stop and answer questions if there are any. Thank you.
Great, thank you, Hidde, for this very, very clear presentation. I think maybe on the last point, I don't know if people like Lily or Lily-Jan or Steve Long want to chime in, because nanobodies have been used especially for structure, to resolve structures of ion channels by cryo-electron microscopy.
So the application that I have in mind is to make nanobodies that can be expressed in the reducing environment of the cytoplasm. Mm-hmm. Not so much for structural biology, but to perturb their function in the context of a living cell. And we have a number of examples of that type. So the nanobodies are unique in that they don't really require a disulfide bond to assume their immunoglobulin-like fold. even in the reducing environment of the cytoplasm, they retain their antigen binding properties. And this makes it possible to create vectors that express in inducible fashion nanobodies against the target of interest. And you can then use that approach instead of mutagenesis to begin to probe functional aspects. So in that case, you wouldn't rely on a mutational analysis of your favorite protein, but rather within the context of a wild-type proteome, you would introduce these perturbants in the form of nanobodies that might occlude one or the other surface of interaction. That's the idea.
That's actually really exciting possibility. We haven't thought about, or we haven't done anything about channels themselves, but even for GFP, there's a group in Germany screening the library and coming up with enhancer and suppressor nanobodies, and explaining at a structural level how they increase or decrease GFP fluorescence. So those, but I guess what's challenging for the beginner is to get hold of just the right library for screening.
So there are synthetic libraries that are probably not quite as good as what you achieve by immunization, but there are companies that will do this on a fee-for-service basis, and one can practice multiplex immunization. So you can immunize one animal with 10, 20 different proteins at the same time, and then screen your library against each individual component of the mixture that went into the animal. But I'm particularly intrigued by the fact that the structural biologists who've done crystallography on ion channels obviously have enough protein for crystallography, which means they also have enough protein for immunization. And to my knowledge, the use of nanobodies for functional perturbation remains to be explored by the channel biologists.
I could chime in a little bit. We've used antibodies to interrogate function of bestrophin and other ion channels. Our main problem is we don't have the fuzzy animals.
That's a problem that can be solved.
Yeah. So yeah.
But I'd be very interested now this is an area where conventional antibodies simply won't do. You cannot express an intact 2-chain immunoglobulin inside a cell. But nanobodies, you can. And you can turn them into what some people call chromobodies by making GFP fusions. Like I said, you can fuse them to any other protein of interest and enforce juxtaposition of proteins that would not normally talk to each other. Stabilize interactions that would be transient. There's lots of things that one can do.
Yeah, I agree. It's very powerful.
So if these proteins are available, say, in a few hundred micrograms, then that's enough to start an immunization and screening campaign.
Maybe I want to move on and give the participants or ask a couple of the participants questions. So one of the interfaces that has between the innate and adaptive immune system that has really emerged in the last couple of years is memory innate immune responses. And one of the questions is whether you can briefly comment on that feature.
Yeah, my understanding of that aspect of innate immunity is that exposure to some of the ligands for innate receptors activates an epigenetic program that sort of permanently imposes a regulatory network on cells that experience that stimulus, such that upon re-exposure, even to an unrelated stimulus, they may respond more forcefully. So my understanding is that this is controlled at the level of epigenetics.
Okay, another question, and maybe coming back one more time to the nanobodies, is that and Kevin Foskett commented that many ion channels lack extensive extracellular domains for binding to nanobodies, but you're talking about intracellular domains of ion channels, which are much more extensive in many cases. Just thinking of LRRC8A, the structure that Steve Long showed, right, which has this huge repetitive leucine-rich repeat domains. But
members of the LRCC8 family form hetero oligomers.
Exactly.
And so the ability to interfere in that heterooligomerization process would be one. The other aspect not to be overlooked is the fact that these nanobodies have an unusual convex binding site that allows them to protrude into clefts where conventional antibodies cannot penetrate. So the exit and entry pores of these channels that were shown so beautifully in Steve's talk would be ideal targets for binding of a nanobody to occlude either the entry or exit phase of an ion channel without otherwise changing its properties. So I'd be very curious to see A, whether this is possible, and B, what the impact of such tools could be.
One question by Asra Malik was that nanobodies are still large. How do they actually get into cells, into the cytoplasm? Are they
You express them minus their signal peptide using lentiviral or retroviral expression constructs. Okay. So this is all done by transfection. There are, of course, claims that by electroporation, you can introduce proteins across the plasma membrane in much the same way as DNA is introduced. I think efforts along those lines have not met with uniform success.
One little discussion that we had in the Q&A chat is related to an earlier part of the lecture, and that is about the activation of the NLRP3 inflammasome by potassium efflux, either through P2X7 channels or other pores. And I think in the field, it's still controversial, actually, how the inflammasome is activated by increasing concentrations of potassium, or even more general, whether there is in fact a reduction in the intracellular potassium concentration, which is based on the on the amount of potassium that's effluxed, and then also the chloride that follows the potassium efflux, and that just leads to cell shrinkage, as one of our participants pointed out.
Yeah.
So I think it's, do you have any particular insights or comments on how this may work, the potassium?
I think Jürgen Rüland in München is one of the experts in this area. I think he is rather enamored with this reduced potassium concentration hypothesis, but to my knowledge, no one has figured out the exact molecular mechanism by which NLRP3 is fully activated.
Yeah. Maybe one of our there are also other potassium channels. I know that Asra Malik, who recently gave a talk at NYU, identified a potassium channel called TRIC2, and there are many other potassium efflux mechanisms that could potentially affect inflammasome activation, but it's how this works, unless you have any insights.
I have no insights there.
Yes, exactly. But this is actually an interesting area of research. And I thank you again for this very, very clear talk, introduction to the immune system. It couldn't be done better.